Hi everyone. We will wait another, I think 3 more minutes and then we'll start. We started chapter 13, right? Okay. Hey Sergio. Sergio, how are you? I can't hear you. Unmute your audio. I do not see Lorden. I see Kaden is here. Kaden, how are you? Kaden? I see Dustin. Dustin? Dustin?
Hey, Ms. Mandy.
How are you?
I'm good. How you doing?
Okay, kind of. And where is Justin?
I don't know.
You don't know?
I don't know.
Okay. So is everything going okay?
Yeah, everything's good over here.
Good to hear.
Hello. Can you hear me now, Nandi?
Hmm?
Can you hear me now? This is Caden.
What? I couldn't hear.
Is it working now?
Yeah.
Okay, there we go.
Okay. What's the time? Okay, let's start now. Okay, I think I talked about this slide, right?
Yes.
Okay, so should I start from here? Second slide?
Disease control, prevention.
Yes.
Yeah, okay, so let's start from here. Okay, so biological safety levels. So the Center for Disease Control, CDC, and National Institute of Health, NIH, have established 4 classification levels called biological safety levels. Each level is designed to protect laboratory personnel and the community. So basically, the people who works in different laboratory, mostly for them, and of course, you know, we are associated with them as a community, so for the community too. Now these BSLs, or you can say the safety level, is determined by 3 factors. 1, agent's infectivity. 2, ease of transmission, and potential disease severity. That's the third one. Okay, so agent's infectivity, the first one, or bacterial or viral infectivity. Okay? How infectious the bacteria is. So if we compare coronavirus and flu virus, okay. So of course, coronavirus is much more infectious than the flu virus. So the degree of precautions that we must need to take for corona and for flu would be completely different. For in this case, for coronavirus case, it would be we have to be much more careful than the flu virus. Oh, not for the flu virus. Okay. Now the second is of transmission. So how easily it can spread? Okay, so coronavirus, so based on that, like ease of transmission, both are almost at same level. Then the third one, the potential disease severity, like is capable of doing epidemic or endemic. So in that regard, the coronavirus is much more dangerous than the flu virus. Okay, so whatever the precautions we need to take for the flu virus. So let's say I'm working to make flu vaccine, flu virus vaccine. And the Dustin is working to make that coronavirus vaccine. So for me, whatever the precautions I need to take for Dustin, it would be much more. Okay, the laboratory should be completely different than the flu virus laboratory, and then all other precaution should be there. Okay. So based on all these 3 criteria and based on the severity of the agent's infectivity, how it transmits from person to person or person to animal and all those, and the potential disease severity, we have 4 basic level. Okay, this is the combined effect of CDC and NIH. Okay, so you can see the pyramid over here. BSL-1, the first level, the second BSL-1, second, third, and fourth. So as you go higher, it indicates high risk microbes, and the bottom, the base of the pyramid, indicates low risk microbes. But at the same time, remember that low risk microbes, they are high in number. Okay. So that's how we make this pyramid. So the first one, our BSL level 1. So microbes are not known to cause disease in healthy hosts and pose minimal risk to workers and the environment. So basically the lowest risk of microbes Um, and for the infection, we have to take a fewest precaution. So these bacteria, like BSL-1 bacteria, who are they? They are who do not actually cause the infection in healthy animal. Number one. Number two, um, um, animal viruses who do not infect, um, uh, human. Number two. Number three, non-infectious bacteria like E. coli, then like B sub. These are the agent for BSL-1. Now, what type of precautions we have to take? So it's the precautions what you take for our regular microbiology lab, exactly almost similar precaution you have to take, like regular, you have to practice that regular aseptic technique. Then you must have all those personal protective equipments like goggles, lab coat. Then there must be a sink inside the lab to wash your hand, and basically all the regular precaution you take in regular lab, biology lab or chemistry lab, biology lab specifically. So all those precautions should be there for this category microbes. Now for the number two, they are indigenous. Okay, indigenous means they are originated in and characteristics of a particular region or country. So basically, you can say they are native or innate microbes. Okay. So in that case, for this group of bacteria, the first level of precautions, like for BSL-1 precaution, that should be there. Plus, um, um, like there must be inside the lab, there must be some extra things like autoclave. That's the next must one. One autoclave machine should be there. Okay, because these organisms are associated with disease of varying severity. So over here they really do not gives the infection, but over here, yes, up to certain level they are. So like mumps, measles, hepatitis virus. So Staphylococcus aureus, you can say mumps, measles, hepatitis. So all those goes under BSL-2. So once you are working against all these bacteria, so all the test tube, plates, and all other instrument tools that you are using, all those must need to be autoclaved. So autoclave machine is very important for this group of bacteria. Then another one is biological septitables. It's called bio. It's called the biosepti cabinets. Okay, that biosepti cabinets, they are actually ventilated, enclosed workspace, you can say. Okay, so basically what happened, it's a desk with wood. Air cannot come out from this cabinet, and we really do not circulate the air inside of this cabinet. We use that HEPA filter. So all the air, we do not reuse air, must need to go out. Out does not mean in the environment, in the open air. It must need to go somewhere where we can actually clean or disinfect this area. Okay, so that's the biosafety biological safety cabinet. Then another one is that shower station should be there. Then eye wash station should be there. All those things, in addition to BSL-1 precautions, should be there for this group of bacteria. Now the next three are indigenous or exotic, means they came from outside, like coronavirus came from China and caused serious or potentially lethal disease through respiratory transmission. Okay, so this group of bacteria are highly pathogenic, and they are lethal through respiratory transmission, like Mycobacterium tuberculosis, you can say the anthrax, okay, Bacillus anthracis, all those group of bacteria. Now you can understand that this group of bacteria are highly lethal, and if you consider the agent's infectivity, highly infectious, transmission very easy because they are respiratory pathogen, so very easily they can transmit. And the potential disease severity, yes, this group of bacteria can have power to go for endemic. They can create that endemic situation like coronavirus, what we are facing now. Okay, so for this lab, this category, for this category of pathogen, the workstation or lab should be a very specific and restricted area. The people who are allowed to go, that that access inside the lab should be very controlled, like not all the people can enter. The people who actually can enter, they must be vaccinated with all, if possible, with all these vaccine of these microorganisms. Then that self-closing door should be there. So two-way, it's called two-way self-closing door, like so automatic door. So you are not going to touch anything. You are just going to stand, it will open or it will close. Exactly same way the ice station and any sink area where that you need to use, that should be also automatic. Person should not touch anything over there. It should be under surveillance, under medical surveillance. Okay, so and as I told that it must be the access must be restricted. Now for this group of bacteria, so these are for BSL-3. So this group of bacteria, the safety precautions for BSL-2 and BSL level 1 must be there. Okay, so BSL-2 and BSL-1 is there. Along with these two, these are the extra precautions that we must have to have for this group of bacteria: Mycobacterium tuberculosis, anthrax, you can say West Nile virus. Yeah, that's another very dangerous. So now the next level, BSL-4, microbes are dangerous and exotic, posing a high risk of aerosol transmitted infections like coronavirus, Ebola, Marburg, which are frequently fatal without treatment or vaccines. A few labs are at this level. In US, we just have 1 or 2 labs for this group of microorganisms, bacteria or viruses. At this moment, I think bacteria are under control, but virus, they are beyond control. So for this, that change of clothing, full body protection, all those things should be there. Like whatever they are wearing inside that lab, that must stay inside the lab. Or maybe over there should be a cleaning area. Usually in this lab, they really do not, with their own clothes, they use that protecting suit. They have special type of clothing system that they use over there. And all of these level of precautions they must take. Okay. Okay, so for this lab, for this type of organism, um, uh, all these level of precautions plus very extra precautions they must take. And as I told that in US we just have 1-2 labs, and they are far away from the locality. No people should live nearby, so all those things are there. So this chart is very important. I am going to give at least 3-4 questions from this BSL level, because as a microbiologist you really need to remember all these precautions, okay, and all these pathogens. So this is a short version, but inside your book, you really need to work on all these 4 levels. Okay, so make sure that this slide goes under BSL level 4 importance. Okay, that much importance you have to take for this slide. This is an example of protective suit that they use at inside the BSL level 4. So yeah, this has its own air supply and maintains a positive pressure relative to the outside, so that if a leak occurs, air will flow out of the suit, not inside, not into it. Okay, so from inside to out, air can go, but from outside no air can enter. Okay, so this is the biological safety cabinet. All these people, they use that respirator. This is the respirator, a special type of respiratory machines. Basically they cannot even inhale the air of this room. Okay, so they have their own respirator. It has fresh oxygenated air, and the air inside of this safety cabinet, we take it outside into a special room or special area, and we disinfect that area. We do not recirculate the air of this room. Okay. So common practices to control the microbial growth. How we can control the microbial growth? So 2 things we have to consider. One is non-living organisms, like the few chemicals or physical methods for the fomites, and for the living organism. So for the fomites, we use disinfectant, sanitizer, and sterilizer. So disinfectant reduces or destroys microbial load of an inanimate item through application of heat or antimicrobial chemicals. So it reduces, it does not kill. Basically, it inhibits the growth. And that's how we can control the growth. So cleaning surfaces like lab, benches, clinical surfaces, bathrooms. When we clean all these area with chlorine bleach, phenol, Lysol, defined type of alcohol. So that's those are the example of disinfection. Sanitization means basically lowering microbial counts on mostly the eating surfaces and utensils, and you can say the area basically safe public health levels, basically public health area, we can sanitize, like eating utensils, cleaning public restrooms, and all those are goes under sterilization. Again, reducing or inhibiting the microbes we cannot kill. So for this purpose we use that phosphates finish, industrial strength cleaners containing quaternary ammonium, that quartz compounds are there, we use to sanitize. Sterilization, so these two only inhibit the growth, okay, we cannot kill. Sterilization, we completely kill the bacteria, so basically vegetative cells plus the endospore, okay, completely. So for these two, we cannot bring down the growth into 0%, but by sterilization, yes, we can bring it down up to 0%. So completely eliminates all vegetative cells plus the endospores and the viruses. And make sure you remember that these are for the fomites, for non-living organisms. Preparation of surgical equipment and the needles used for injection, all those things goes under sterilization to control the infection and not to spread the infection. Pressurized steam, basically autoclave and different types of chemicals are there. We used the ionizing radiation, not the non-ionizing, only the ionizing radiation we use to sterilize. Now for living, we can apply antiseptic. So reduces microbial load on skin or tissue. So again, it inhibits the growth, but we cannot bring it down all the way up to 0. Boric acid, isopropyl alcohol, hydrogen peroxide, iodine, all are example. Degerming means removing microbes from a limited area. And degerming mostly results by mechanical removing, okay, like wiping the area with bleach, alcohol, like when we apply alcohol before injections, all those goes under degerming. Hand washing, another example of degerming. As I told, that you are physically controlling the microbial growth. And soap, alcohol swab, all those are example, all those we use to degerming. So measuring microbial control. So we use all those things like physical chemical processes. How are we going to measure that Lysol is working, okay, versus Listerine is working? So how are we going to measure? So we have two terminology we frequently use to refer the effectivity of the reagent. One is the suffix sidal or side we use, and another one is stat or static. So when we use any word that has the suffix side or sidal, that means the type of microbe or infectious agent killed by the treatment. We can kill the microbes. So basically when it is bactericidal or bactericides, means that that agent can kill that bacteria. Virucidal or viricides means that agent can kill the virus. When it is stat or static, means it cannot completely killed, but it can have control over the growth. So basically it can inhibit the growth or it can stop their growth. Okay, so bacteriostatic means it inhibit the growth of bacteria. Fungistatic means it can inhibit the fungal growth. Factors that determine whether a particular treatment is sidal or static include the types of microorganisms targeted, the concentration of the chemical used, and the nature of the treatment applied. So before even we talk about, okay, so it is bactericidal, you have to consider three things, okay, like types of microorganism. So when you are talking about, okay, so it is bactericidal, then consider, is it a vegetative cell, or you are targeting the endospore. Okay, so let's say it's 70% alcohol. Now 70% alcohol is bactericidal. So it is bactericidal, sorry, it is bactericidal for vegetative cell. So it is cidal for vegetative cell, but it could be static for endospore. It's not cidal for endospore. 70% alcohol might not kill the endospore. So your target should be considered first. Then the second, the concentration of the chemical used. Okay, how much you are, what's the percentage of your chemical agent? That of course very important to measuring the microbial growth. Are you using 70% alcohol, 30% of alcohol, or 100%? Of course, the effect of 30% and 70% alcohol will be completely different, okay, will be variable. So that you need to consider. And the nature of the treatment applied. What type of treatment you are applying? Is it a chemical agent, physical agent? What are you going to do? So all these things, three things need to be considered. So how are we going to measure that what's the effect of, to determine the effectiveness of any control method? It could be a physical agent, it could be a chemical agent, whatever. How are we going to measure? Like let's say it is autoclave, okay, versus if it is 70% alcohol. So how are we going to measure that which one is effective against let's say coronavirus? So how are we going to measure? Are we going to count the number and say, okay, so autoclave machine killed 10,000 viruses, and 70% alcohol killed only 100 viruses? So are we going to go behind the number? Are we, or maybe the percentage? So how are we going to measure? Okay, so you can understand when I'm going behind the number, that autoclave machines, okay, so how much time it took to kill all 10,000 virus to kill? Okay, so that question will arise. Now what was the temperature over here, what was the pressure over here? Versus 70% alcohol, it just killed 100 viruses. So how long it took to kill the 100 viruses? Now next question, okay, so if we add that pressure and temperature that we have used during autoclave, so what effects will be here for the 70% alcohol? So all those questions will arise. So it really does not make sense that just talking about the number or just the percentage. So what we do, degree of microbial control can be evaluated using a microbial depth curve to describe the progress and effectiveness of a particular protocol. So usually we use a graph by plotting the time on one side and the number of cells on another side. So x-axis we have time, y-axis we keep all the number, Okay, so it becomes easier to find out the logarithmic effect of the microbial control. So bacterial population die at constant rate as rate of killings remains constant even when the population size varies. So what we look, we actually look for the decimal reduction times. Okay, so basically to control, to measure the effectiveness of any agent, controlling agent, we take help from the graph, because when we measure the effectiveness, we go behind the percent of killing. Population size actually does not matter. So we are interested about the what is the rate of killing of that microorganism, and every time that population die at constant rate as rate of killings remain constant, even the population size varies. So let's say 70% alcohol. 70% alcohol can kill, let's say, 3 bacteria. The area does not matter. Okay, so they can kill just 3 bacteria. So and the number matters over here, and just say, let's say just 0.1 microliter of 70% alcohol can kill 3 bacteria. Now that amount of bacteria you can spread over here, you can spread the whole area over here, you can spread the whole area over here. As I told, that size does not matter, just the number matters. Okay, why? Because that's the power of that 70% alcohol. This amount of 70% alcohol can kill that bacteria. Okay, so that 70% alcohol, it does not matter we have 10,000 bacteria or 20,000 bacteria or 1 million bacteria over here. So population size does not varies, and it's not like that. Okay, so at the beginning we had 10,000 bacteria, so it just 70% alcohol killed only killed 3 bacteria, but now we just have 50 bacteria left, because already 70% alcohol killed all, we applied all, to kill all bacteria, so only 50 bacteria are there. Okay, so now if we apply that to 1 microliter 70% alcohol, it will still going to kill the 3 bacteria. It does not matter the size of the population. It's not like that. Okay, so when it was 10,000, it killed 3 at a time. Now it's 50, so it is going to kill 1 bacteria at a time. No. Okay, so bacterial population die at constant rate. All these agent that we use, it's true for all. Okay, and that's how we actually prepare all of our agents, controlling agents. So the percent killed is more useful than the absolute number. So over here, if we tell that, okay, 70% alcohol killed 5,000 bacteria, does it make sense? No, it does. It really does not make sense. Okay, so absolute number does not matter. So over here, we go by the percent of killing. So basically, the rate of killing. Okay, so that to kill 3 bacteria, it just took 1 minute or 1 second. And what is the strength? It is 70% alcohol and just 0.1 microliter. So decimal reduction time is the time it takes to kill 90% of the population when exposed to a specific microbial control protocol. So how we measure? We go by the decimal point, or decimal reduction time, or it's called D value. D value is the time it takes to kill 90% of the population. So we started with 100 bacteria, okay, when it was 9 a.m. And now we have only 10 bacteria, and it is 9:10 a.m. So basically, to kill all 90 bacteria, it took 10 minutes. Okay, so that's our D value. So now we are going to work on the physical method. So we have how we actually now control. So till now we talked about the agent, controlling agent. So we have physical controlling agent, we have chemical controlling agent. So at this moment we are going to work on the physical, you can say, methods. Okay, so dry heat sterilization, so heat. Okay, so first one is heat, and we are working on dry heat sterilization. Dry heat sterilization, like you sterilized a loop by using the bacteria incinerator you used, in the lab. You heated up the lip of the tube. That actually not sterilization, but you did this one to sterilize. So that goes under dry heat sterilization. Okay. Now, you have to consider two things: thermal death point, TDP, and the thermal death time. So when we're talking about thermal death point, we are actually working for the temperature. So thermal death point of a microorganism is the lowest temperature at which all microbes are killed in a 10-minute exposure. Okay, so time over here, time is 10 minutes. We are killing 100% of the microbes, and we are looking for that temperature. So that's the thermal death point. Thermal death time, over here, the length of time needed to kill all microorganisms in a sample at a given temperature. Okay, so length of time. So how long it took, that's what we are looking for. Over here, we are killing 100% microorganism, and we know the temperature. Okay, we know the standard temperature over here. Autoclave, another applying heat, but in this case, this is an example of moist heat. Okay, so for moist heat, we use autoclave machine. So we have one autoclave, large autoclave machine in our lab. I thought I will show you, but anyway. Okay, so this is the chamber. This is the main chamber. What we do, we take out all the air from the chamber. So this chamber is air free, and all these tubes or rods, hollow rods, they are we supply steam through this rod. Now, when there is no air and we have steam, we filled out this chamber with the steam. That means we create an environment where the temperature is more than 100 degrees centigrade. Okay, if we boil water, we can reach all the way up to 100 degrees centigrade. Okay, so that that is very easy. That temperature, to get that, to achieve that temperature, that's very easy. But it's not enough for endospore and other bacteria there. We cannot kill those bacteria. We must have more than 100 degrees centigrade. Now, how are we going to get that temperature? Because that's our maximum limit. Okay, so steam, in that case, steam is the solution, because I hope you understand that 100 degree, at 100 degrees centigrade temperature, the temperature of water and temperature of steam, that actually varies. Steam has more temperature. So in that case, so when we are supplying steam, we can reach all the way up to 121 degrees centigrade. Okay, and that is good for endospore. Now, as I told that we take out air and we are supplying steam, means we are increasing pressure. In autoclave, we usually go for 15 to 20 PSI. That much pressure we can create. And this 121 degrees centigrade and this high pressure is actually very helpful to kill all the endospore. That level of cleaning we can killing we can do over here. Okay, and that's an example of moist heat treatment. Pasteurization, what we do, we actually treat it is usually for the food materials, because um, we really want to preserve the food quality and all the vitamins, minerals, hormones, enzymes associated that we get from that food. Okay, and if we boil, let's say the milk, if we boil, then we are actually going to lose all the food value thing. Okay, most of the vitamins, enzyme will be denatured. So to avoid that thing, but at the same time we cannot use even raw milk. Okay, we have to increase the shelf life. We really need to keep it for the longer time. So we do pasteurization for that. Now pasteurization, what we do, we treat that item with certain temperature, but of course we maintain the temperature so that we can maintain the food value. Okay, so we cannot boil, we don't take the temperature all the way up to the 100 degrees centigrade, and to do that we have to boil for 10 minutes. We don't do that. Okay, so our target should be for pasteurization, so low temperature, but at the same time keep the food value same. So that's a pasteurization. So when it is low temperature, means we cannot kill all the microorganism. Okay, what we do, we actually keep that number low. Okay, so that number remains lower than the usual, and that number is usually not harmful for our health. And certainly we try to kill some of the pathogenic from there. So that's the pasteurization. Basically, what we do, we inhibit the growth, or you can say we try to slowing down the metabolism. Okay. We have two different type of pasteurization process: high temperature short time and ultra high temperature. So when it is HTST, high temperature short time, milk heated at 72 degrees centigrade for 15 seconds. Low temperature, longer time. For ultra high temperature at 138 degrees centigrade for very short period of time, only for 2 seconds. We keep the milk only for 2 seconds at 138 degrees centigrade, more than boiling water. Okay, but for boiling water, you have to keep boiling for 10 minutes. Okay, so over here, the temperature is much higher than the boiling, but we only keep it for 2 minutes, and that is effective. Refrigeration and freezing. So basically our regular refrigerator at home, lab, they actually maintain temperature from 0 to 7 degrees centigrade temperature. Okay, this is not the temperature where we can kill. No, we cannot kill, but we certainly slow down the metabolism, and that means we can inhibit the growth. And what is our target? Can we completely inhibit the growth? No. Our target is we are going to keep that product or store that product for short period of time. Okay, these are not for mean to keep a long life. So can we keep the milk 1 year inside the refrigerator? No, but we certainly can keep that one for 15 days up to 30 days. That we can keep. Okay, so this is our target. If the temperature goes below -2 degrees centigrade, they can stop the microbial growth. So over here, when the temperature is varying from 0 to 7, it only can slow the metabolism or inhibit the growth. But if we pull down the temperature all the way up to below -2, then it actually can stop the microbial growth. For long-term storage, we use that. We have that ultra low freezer where the temperature we maintain below -70 degrees centigrade for different types of vitamins, vaccines, like now the China, they have that coronavirus. I'm not sure, but if they have that coronavirus vaccine, okay, so for use, future use for the whole world, they must need to use that ultra low freezer. In US and all other countries, whoever is trying to make that vaccine, they all must need to use that ultra low freezer. Okay. Over here, we pull down the temperature all the way up to below actually -70, and for that we use either the dry ice or liquid nitrogen. And you can understand these are for the long-term storage or for the transportation. If China is going to send us the vaccine to US, so they must use that while transporting that material. Pressurization, we actually, by using the pressure, I have told you that for the autoclave, what we did, how we increases the pressure. So pressure is a very important agent to control microbial growth. So we can actually kill the microbes. Pascalization means we use the high pressure in food industry. So in food industry, we use that pascalization. We use that canning. So when we use at home, when we use to make that the jam, jelly, to store, that's the canning. We use that pressure. So 100 to 800 megapascal is sufficient to kill vegetative cells by protein denaturation. Endospore can survive. This much pressure, when we do canning, we can create this much pressure to kill the vegetative cell, but endospore remains there. So when you do this canning thing at home, that for the Clostridium and other bacteria who can make spore, very frequently, all those cans, food cans, they actually store that Clostridium endospores. Okay, so you have to be very careful for that. Hyperbaric oxygen therapy, we use pure oxygen with a high pressure than the normal atmospheric pressure to treat certain diseases, like the patients who are hypoxic. Hypoxic means their cells and tissue, inside their cells and tissue has low oxygen. So basically their body cannot supply enough oxygen into their cell, and especially if there is not enough oxygen at brain, the stroke and all those things can happen. So to treat that hypoxic condition, there are people who has asthma, cancer. For all those people, we use that hyperbaric oxygen therapy. So what we do, we actually keep that patient in a chamber. That chamber maintains high pressure, and that actually supply. It feels really good. You will look fresh and really hyperoxic, energetic. Your skin will glow. All those things. It's nowadays it is becoming a very popular therapy. Desiccation, um, for long time we are using these are very old process like sun drying. We keep, uh, we actually do that like to dry up the tomatoes, resin, and all those. Um, Fish. Um, we also use certain type of chemicals like salt, and basically by using the chemicals we want to create one hypertonic environment, and you know what happens when it is hypertonic solution. Okay, so hypertonic means salt, more salt, less water. Inside we have more water, so water goes out, and all the cells becomes dehydrated, and that's how we take out all the water. How actually we do the sun drying. So what is our target? All the water they evaporate from the cell with that UV rays and the high temperature. Radiation, we have two different type: non-ionizing and ionizing radiation. So in lab, we actually worked on UV radiation. Remember when we did that physical control lab? We kept all the dishes, petri dishes under that UV machines over here. That's the non-ionizing. In case of non-ionizing ray, like UV, that's an example. What happens? This ray actually create thymine dimer. This thymine dimer means they make hydrogen bond in between two thymine. So when DNA polymer is going to read during DNA replication, when it try to read, mistakes happens because it cannot go. So if it is going reading by this way, this is the polymerase. If it is going this way, it gets stuck over here because it cannot proceed because we have a hydrogen bond, unusual hydrogen bond over here. And if it jumps from here to here, in that case, it creates mutations, false base pairing, and mutation occurs, and basically that's how it can inhibit, the bacterial growth. And you have seen that we did, one is open, we made three different types of plate. One was open, one was closed, and on top of one it was open, but we covered that with one cutout paper. I hope you remember. So, and what happened? So basically this area, the cutout area over there, we had enough growth because this sorry, no, not enough growth because they cannot go through all these paper cutouts. Okay, so we had no growth over there, and we had enough growth over here, this area. Now, ionizing radiation like X-ray, gamma rays, they are much more powerful than the UV. They can enter into the cell and alters its molecular structure. They can even move through the plastic, paper, and all those. So nowadays we use, to uninfect or disinfect the plastic products and paper boxes and all those. We use this ionizing radiation, and even the food. Now there are few countries like Australia, New Zealand, Netherlands, they all use these ionizing radiation to clean up all the foods. But over here, not only here in US, but throughout the world, there are different countries, there are different people who are still very scared to use this type of product. So over here, um, that FDA, they actually uh, made some rule that irradiated foods must be clearly labeled and displayed. So if people wants to buy, it's their choice. The radiation symbol, it's called radura. Okay, so if you see this symbol, that means your apple has been disinfected with ionizing radiation, maybe X-ray. Usually we use the gamma rays to do that, but scientifically, if you ask me, it is completely safe, because that gamma rays does not change your apple or your vegetable or your fruits. Okay, so you can basically, the people really need to know the science behind the topic. Before even we know, before even we do read, we actually talk about it. So that create the confusion, but it is actually safe. Um, in this case, you are going to lose little bit of vitamins and all those, but it's much better than taking all those chemical pesticides, chemical agents and all those, when apple is cleaning with some types of hormones and some types of chemical, that the residues remain, and the upper layer of the skin, they absorb, and you are taking that, so it's much better. I think this ionizing, that gamma irradiated foods are much better than the other stuffs available and we use. Sonication, in this case, we actually use ultrasound. Okay, we have a machine, it's called sonicator, and by used ultrasound, we actually disrupt the cell structure. That sound wave actually can breaks down the cell membrane. HEPA filter, what happens over here, that filter sheet, it has a pore that much smaller. I think it's much smaller than 3 millimeter. So this pore, it's like straining. Okay, so all the virus, bacteria, small bacteria, large bacteria, virus who are larger than this pore size, they get stuck. So when you change the filter paper, that means you actually throwing out all those virus, bacteria, and when they are stuck inside that pore, they become inactivate anyway. Another type of filtration, membrane filter, it is much stronger than the HEPA filter. I hope you know that HEPA filter we used at clinical settings, car, at home, airplane, biological safety cabinets, everywhere we use that HEPA filter. Membrane filter is much smaller than HEPA filter. I think it's, I forgot the exact, but I think it's less than 0.2 millimeter, the pore size. Okay, and over here we really do not use any temperature. So without temperature, and because of the small pore, we actually, it's kind of suction, and as a result, it is very, very useful for to filter like vitamins, like antibiotic, vitamin solution, and all those. We usually use that, like no temperature. So all these things, the protein enzyme remains intact. So this is the list, whatever we talked about. Now the chemicals, so we are done with the physical control. We are going to work on the chemical control. Previously, remember that Joseph Lister, I hope you remember, he applied that carbolic acid, okay, first. So till now, I think even 5-10 years ago, we used to use carbolic acid, but it is a skin irritant that means it is phenol actually. Okay, so this is the structure of phenol. This is the benzene ring, and benzene ring is associated with hydroxyl group. Okay, so that's the, so it is benzene ring plus that hydroxyl group together makes one phenol. Make sure you remember the structure of phenol. I can give you the structure of phenol and phenolic acid. So Joseph Lister, he actually discovered that, and he started using that. He didn't discover, but he discovered the process. He started using that carbolic acid before surgery and to disinfect all the surgical instrument. But phenol is very, very skin irritant. That's why now we actually modified it. So it is called phenolics. Phenolics means usually we use that phenol structure and we added something with it. Okay, so phenolics are compounds that have this group as part of their chemical structure. So it carry that phenol structure and few little extra stuffs. You really need to remember these two picture. So phenolics is used instead of carbolic acid, consists of benzene ring with an OH group. This OH group. Phenolics are less toxic than phenol and inhibit microbial growth by denaturing proteins and disrupting the membrane. Some of the phenolics usually we make at lab synthetic, but there are a few we get from the nature, like thymol, eucalyptol, we get it from plant. Phenylphenol, a type of phenolic, has been used as a disinfectant as well as to control the bacterial and fungal growth on harvested citrus fruits. Okay, lots of vegetables and fruits, we actually use these phenolic compounds, you know, to keep it fresh from long time. Hexachlorophenol, and that's what I told, that using this it's better to use that gamma radiated food instead of all this. Hexachlorophenol, another phenol known as it is a bisphenol. It has two rings. Over here is the active ingredient in pH isohex. And triclosan, you all know this one, the triclosan, I think. Triclosan is a common ingredient in antibacterial soaps. We have it in toothpaste. We actually started triclosan in toothpaste first. It is also bisofen by two, you know, these things. Bisphenol, basically two ring structure. Nowadays, like cutting board, knives, all are, we are trying to embed that triclosan. So all those things like cutting board, it will be automatically, you know, microbes free. So we are trying to do that. All the hand soaps, hand wash material all has that triclosan. Heavy metals, there are metals, heavy metals actually denature proteins, impairing cell function, and thus giving them strong and antimicrobial properties. Like copper, if you use that, you know, copper doorknob, door handle, it kills microbes that otherwise might accumulate on frequently touched surface. Eating utensils contain small amounts of silver to inhibit microbial growth. But in this picture, you can see these people is suffering from argyria and irreversible condition caused by bioaccumulation of silver. Too much silver Okay, so copper commonly lines incubators to minimize contamination of cell culture stored inside. Antiseptic mouthwash has zinc, okay, heavy metal. So if you use up to certain um, percentage, they are good, but overuse, you can see the effect. Halogens, iodine, chlorine, and fluorine. So they are chemically not that active because they are fulfilled their outer um, electron, with 8. So betadine is a solution of the iodophore povidone iodine. It is commonly used as a topical antiseptic before surgery. We still use fluorine. You can must have inside your toothpaste. Okay, at certain percentage. Too much fluorine is carcinogenic. Chlorine, all the disinfectant, we must have chlorine. Alcohols, ethyl alcohol, the intoxicating ingredient found in alcoholic drinks, is also used commonly as a disinfectant. Isopropyl alcohol, that rubbing alcohol, another commonly used disinfectant. So it's well known. Soaps, now the soaps are the salts, okay? So basically salts means they are the combination of acid and alcohol. You can say acid and base. That gives us salt. So they has both the acidic part and the basic part. Okay, so one hydrophilic head and one hydrophobic tail they have. So so salts or fatty acids and have the ability to emulsify the lipids, fats, and oils by interacting with the water through their hydrophilic heads and with the lipid at their hydrophobic tails. So just try to imagine for, let's say it is gram negative bacteria. Okay, so they have one outer membrane, and over here they have another, no, this is inner membrane, let's say, and this is outer membrane. Okay, and they have peptidoglycan over here. So that soaps actually destroy the lipid, they emulsify the fat. Okay, so basically one lipid, they are broken down into small monomers. So it becomes very effective against that gram negative bacteria. If you can destroy this part, it's easier, and then you can breaks down this part. Surfactants, detergents, we called that quartz, that's a quaternary ammonium salt. What they do, they actually separate all the phospholipids from one another. Okay, so quartz are able to infiltrate the phospholipid plasma membranes of bacterial cells and disrupt their integrity, leading to death of the cells. This is the proper hand washing technique. So get running water, then lather with soap, then scrub for 20 seconds, at least 20 seconds, and then rinse your hand under clean running water and then dry. Disk diffusion method, you already we worked on both on Labster and on antibacterial lab. I'm not going over it. For the disinfectant also we use the same thing. These are the paper disc soaked with antibiotic or with the disinfectants, and we place it on the agar plate above the microbes, and it is going to create that zone of inhibition, and then measure and we can test the effectiveness. Another use, to test the effectiveness, it's called in-use test. Okay, that usually we use at clinical setting. So what we do, this is the disinfectant, and this is the 9 milli sterile broth with inactivator. Okay, so we just transfer 1 milli disinfectant over there, and then almost 0.2 milli or maybe 10 drops we transfer on 2 plates. One we incubate at 37 degrees centigrade, and for 3 days. Another one we incubate for 7 days at room temperature. Any one choice you can have. Growth of 5 more colonies on either plate indicates the contamination of disinfectant solution. That's the in-use test. Usually on the clinical setting, we use this in-use test. Most effectively, we can use this. So we are done. Do you have any question? This chapter is easygoing, not hard, but lots of information that you have to memorize, but it's easy to remember. Most of the things you know. Okay, so do you have any question? No? No question? No? Who is this? Corona Beer? I think it's Joe. Joe?
You
I
like
like
my
the
background?
model. It's nice. Can I take a picture of this model?
Sure. I got it off the internet.
Okay, so I took a picture. Okay, so your third test, okay, it is going to cover chapter 9, 10, 11, 12, 13. It is already there, it is already open, okay, from I think 7 o'clock in the morning or 6 o'clock in the morning, I forgot. It's already open. You can take that test anytime from today till Saturday night 11:59. So it will be closed at 11:59 on Saturday. So you have today's Tuesday, Wednesday, Thursday, Friday, Saturday, almost 4.5 days, more than 4.5 days to take that test.
Nice.
Okay.
Good.
Make sure to work on all the sample question. If you work on the sample question, you should do good. Okay. Now the another problem I am facing, this lecture ends up at 12:15, and my next lab starts at 12:30. Usually I talk more, so in between I do have only 5-6 minutes time, at the max 10 minutes. So to convert this class into video or to get the audio recording, it takes at least 20 to 25 minutes, more than that actually. So that's why I'm not getting any, it's getting very tough for me to get that recorded version of the Zoom session. So do you mind if I post my other lecture? Like I have another class on Monday, Wednesday. Can I post that lecture Zoom
Yes,
session?
ma'am. Yes, ma'am.
Okay,
Sure.
I will do that, because last week also I told that okay, I'm going to post, but I didn't get any. There was not enough time in between my lecture and lab. Okay. Okay, so any question regarding third exam? No? Any question from discussion and infographics?
Yes, for the discussions, I saw there were like 3 different topics. Are we to work on all 3, or you just want us to work on one? What are the clear instructions on that?
No, 2.
2 questions?
And make sure to go over to read the syllabus addendum, the new syllabus. All the detailed information are there. Even you can find out the rubric
Okay.
there. Okay, so before even you start working on, read the syllabus properly. So for the discussion, I gave you 3 topics. You
Yeah.
are supposed to do 2.
Yeah.
So one is extra. You can choose any 2 out of those 3. And now the another thing is like for each topic there are multiple questions, like 1A, 1B, or maybe the question 3. I didn't mark it as A, B, C, but it has multiple question. So you have to answer all.
Okay. Another question is on the new syllabus, under the lecture grade that shows the grade for the lecture. It says discussion is 10 points times 4 is 40 points. How does that happen? I didn't get that.
Okay, so remember that's for 2 tests.
Oh, I'm sorry?
You have exam 3 and exam 4 left, right?
Yes. Yes.
For both of these tests, so exam 3 and exam 4 will go for 70 points, right?
Yeah.
So 70, 70, 140. So what are the left? Means 60 points left, right?
Yes. Yes.
For together the third test and 4th test, means each test needs 30 more points.
Yeah.
Okay? So for both of these tests there will be 4 discussion. Each discussion will carry 10 points, so 4 into 10, that gives me 40, and 2 infographics means 2 into 10, 20. That's how I'm going to recover that 60 points.
So the discussion there is not the discussion topics you're giving us. This is a separate discussion?
No, that's the discussion. So, okay. So basically what you have to do for each test, just consider third test. For third test, you have to take the test for 70 points. Rest of the 30 points you are going to get from 2 discussions and 1 infographic.
Okay.
Okay. So, oh, my video is off. Okay. So what is happening for each test? For third test, you have to work on 2 discussion and 1 infographics to get that 30 points. Again, for the 4th test, you have to do 2 discussion and 1 infographics.
Oh, okay.
So that's why that when you see on the syllabus, so total 4 discussion altogether. For the rest of the semester, actually you have to do 4 discussion, 2 for the 3rd test, 2 for the 4th test.
Oh, okay.
2 infographics, 1 for the 3rd test and 1 for the 4th test.
Okay, I understand now. Thank you.
And these questions aren't, I mean, the discussion's not due until April 30th. Is that going to be for both exam 3 and exam 4?
Yeah.
Okay.
Yeah. I am going to post your next infographics topic and the discussion topic probably by Sunday night.
For exam 4?
For exam 4.
Oh, okay.
Yeah,
And is all
and you can start working and you can go by your own pace.
and you said all the infographics and discussions, so all of them, including for exam 4, is going to be due April 30th?
That's the target date, but I won't be that strict. So if you want to take maybe 2-3 days extra, I will allow. As long as you are all done before final exam, I really need to get a little bit time to grade. Because I forgot, what is your final exam date? Is it on 11th or 5th?
5th.
Fifth. Okay. So for you, as long as you are done by the 4th, you should be fine. I think my other class, their final exam is 11th. I really need to get little bit of time because I think 12th is the last date for the grade. So I have to post your grade positively by afternoon of 12th. So I need a little bit time to grade all those.
Okay.
So as long as you post before your final exam, you should be fine. I will be happy.
Okay.
Okay.
All right.
And for the infographics, Erika, I can see you now.
Hi.
First time.
Yeah, I just had a quick question. My computer's
Mhm.
about to die. But I just looked at the discussion topics. There's only 3 questions.
Mm-hmm.
Is that right?
Yeah, you
But
have to do,
there's-
you have to work on only 2.
Okay. So
So
but there's 3.
these discussion topics only for exam 3. Did you get
Okay.
it? This discussion
I thought we had to choose 2 now and then 2 later from this.
later, yeah, and for that I'm going to give you another sets of questions.
Oh, I understand.
Because that discussion topics will come from your 4th test. These topics are all came from chapter 9, 10, 11, 12 and 13. Because these are the only chapter we are supposed to work on for chapter test 3. So next topic will come from the chapters that you will work for exam 4. Get it? Clear?
Got it.
Yeah.
Yeah.
Okay. Any more questions? Okay. So one more thing I want to clarify about the infographics. Okay. Infographics means you are going to make poster. Okay. So basically, if you are making PowerPoint, make sure it should be on just one slide. Just one slide. If it is canva.com, just one poster. Now, within that poster, you have to have 5 facts and minimum 3 pictures should be there. Okay. So 5 facts will come from the reason why that gram-positive bacteria didn't grow, or if it grew on the slide, why cannot I see that gram-positive bacteria? So did I, if it is the stain problem or what's the problem? I couldn't set up the microscope. So many facts could be there. So try to find out 5 facts and 3 pictures should be there in one poster. Clear? Nico?
Yeah.
Did you get it?
Yes.
Kathy? Are we good?
Yes.
Rashida? Why can't I see you? I really want to see you. Now I can see you. Okay. So did you get it? Are you clear? The infographics?
Yes, ma'am.
All of you?
Yes.
Yes.
Okay. Okay, so take the test and you must have responders and lockdown browser. Oh, I have to start lab. Okay, so bye.